A cascade is a chain reaction of molecular events inside a cell. One activated molecule triggers the next, which triggers another, and so on, like falling dominoes. The purpose of a cascade can be to amplify the signal or to increase the speed and efficiency of the reaction. Proteins are mainly activated through phosphorylation. When a kinase enzyme uses ATP, then the terminal phosphate from ATP is transferred to the protein. Then the protein's shape changes, which exposes the active site, and then the protein becomes functional and a ligand can bind to it. A protein becomes deactivated by removing the phosphate group. A phosphatase enzyme binds to the protein. It removes the phosphate group. Then the protein returns to its original shape, the active site is hidden, and then the protein becomes inactive. A ligand binds to a receptor on the cell membrane. This causes the receptor to change shape, activate. In the second step, the relay proteins are activated. The activator receptor interacts with the first protein inside the cell. Often this involves a protein kinase activated. In the third step, phosphorylation chain reaction, now the real cascade begins. Kinase A becomes activated, then kinase A adds a phosphate group to kinase B. Kinase B becomes activated. Kinase B phosphorylates kinase C becomes activated, and the final activated protein causes a cellular response.
Kinase is a protein that phosphorylates. Tyrosine kinases are proteins that form dimers. They transfer phosphates to relay proteins. At first, 2 receptors receive a ligand each. After each receptor has a ligand, they join to form a dimer. Next, 6 ATP come in and attach to the proteins, leaving a phosphate each. The phosphate is added to relay proteins, activating them. An example of a tyrosine kinase receptor in real life would be an insulin receptor.
A ligand binds to the G protein coupled receptor on the outside of a cell. GDP is exchanged to GTP and the G protein becomes active, and the GTP moves to adenylyl cyclase. Adenylyl cyclase converts ATP into CAMP.
CAMP stands for cyclic adenosine monophosphate. It's a secondary messenger, a small molecule inside the cell that helps carry and amplify signals from the outside to the inside. A hormone or neurotransmitter, like adrenaline, binds to the G protein coupled receptor on the cell membrane. How it works. A hormone or neurotransmitter, like adrenaline, binds to the G protein coupled receptor on the cell membrane. The receptor activates a G protein, which then activates the enzyme adenylate cyclase. Adenylate cyclase converts ATP into CAMP. CAMP activates protein kinase A, which is an enzyme that adds a phosphate group to other proteins. This phosphorylation changes the activity of those proteins, altering cell functions like metabolism, gene expression, or ion channel activity. One hormone molecule can create many CAMP molecules, which then activate many pKa molecules, amplifying the signal inside the cell.
Epinephrine response occurs in 6 steps. In step 1, epinephrine binds to the receptor, which causes the receptor to change shape and become active. In step 2, the G protein swaps GDP for GTP, turning it on. In step 3, adenylate cyclase is activated. The active G protein turns on the enzyme adenylate cyclase. In step 4, CAMP is produced. Adenylate cyclase converts ATP into CAMP, cyclic AMP. CAMP acts as a second messenger that spreads the signal through the cell. In step 5, protein kinase A, pKa, is activated. This happens when CAMP activates protein kinase A. pKa starts a phosphorylation cascade. In liver cells and muscle cells, pKa activates enzymes that break glycogen down into glucose and inhibit glycogen synthesis. The result is increased blood glucose. In heart cells, pKa phosphorylates proteins that increase calcium availability and strengthen the speed of heart contractions, which results in increased heart rate and force. In lung and airway muscles, PKA causes muscle relaxation, which results in bronchodilation or wider airways, which kind of encapsulates the flight or fight response.
The ligand binds to it, the receptor changes shape and opens. Once the receptor changes shape and opens, it allows ions to flow in and out of the cell, changing its charge. This change is called depolarization, which can trigger an action potential in neurons and contraction in muscle cells. An example is the AMPA receptor in the brain, which is a type of glutamine-gated ion channel.